Hello students, welcome back. This is Kiran Kumar, NEET Foundation faculty, and today we are going to start the next topic, changes in classification system, from the second chapter, biological classification. So this topic is going to be very important. So listen carefully. Let's start. So in the last class we have discussed different systems of classification, such as your 2-kingdom classification, 3-kingdom classification, 4-kingdom classification, 5-kingdom classification, which was proposed by R.H. Whittaker, and the 6-kingdom classification proposed by Carl Woese. Correct? So let us see what changes took place in the classification system regarding the position of different organisms. So here, earlier classification. In earlier classification, the taxonomists followed the 2-kingdom classification proposed by Carolus Linnaeus, that is, only 2 kingdoms were studied, that is, kingdom Plantae and kingdom Animalia. Afterwards, in 5-kingdom classification of Whittaker, some changes have took place. Some of the organisms were removed from plant kingdom and were placed in their appropriate kingdoms. Okay, now let us see what changes happened. Next, in earlier classification system, the kingdom Plantae, listen carefully, this is very important. The kingdom Plantae, it included bacteria, it also included blue-green algae, It also included fungi. Even fungi were also included under plant kingdom, and it also included ferns. It also included angiosperms. It also included gymnosperms. Okay, so look here, bacteria, even though this bacteria are not plants, they were placed under kingdom Plantae. Why? What is the reason? Because the bacteria has cell wall. So that is the reason bacteria were placed in the plant kingdom. And blue-green algae. What are these blue-green algae? These are nothing but cyanobacteria. They are also one kind of bacteria which are capable to carry out photosynthesis. Even these people also got cell wall. That's why they were included under plant kingdom. Next, fungi. Fungi are heterotrophic multicellular organisms. Some are saprophytes and some are parasites, which we have discussed in the last class. Fungi cannot prepare their own food, but even though the fungi were placed under plant kingdom, why? Because they also have cell wall, which is a common character. And ferns, you know, these are a kind of plants which does not produce seeds. Angiosperms are the plants which produce seeds inside the fruits. Gymnosperms are the plants which produce seeds directly without fruits. That means the seeds will not be enclosed inside the fruits. So all these people were included under plant kingdom. Okay, so the plant kingdom also included unicellular algae. Chlorella was also placed under plant kingdom in a group called as algae, and another unicellular motile algae called Chlamydomonas. This was also placed under algae of plant kingdom. And another filamentous algae, Spirogyra. It is also a filamentous algae. So this is multicellular organism, and these two are unicellular organisms. All these 3 people were included under a group called as algae of plant kingdom. Okay, so this used to be the trend in the earlier system of classification. So after R.H. Whittaker proposed the 5-kingdom classification, the criteria of classification has changed. So the organisms earlier which were placed under plant kingdom, they have to move to their respective kingdoms. Similarly, in kingdom Animalia, it included all the animals and also your unicellular organisms Amoeba was also included under animal kingdom. Why? Why amoeba was placed under Animalia? Because it doesn't have cell wall. It lacks cell wall. Even paramecium, which is a unicellular organism, this was also placed under kingdom Animalia. So all these people, they used to be the members of kingdom Animalia. And here, bacteria, blue-green algae, fungi, ferns, angiosperms, gymnosperms, all these people were kept under kingdom Plantae. Now let's see what changed in Whittakers classification. In Whittakers classification, the bacteria and the blue-green algae, they have to be relocated to new kingdom, that is kingdom Monera. So a new kingdom has been created for these two guys. So they were kept under kingdom Monera. So bacteria were moved under kingdom Monera, and blue-green algae, that is a photosynthetic algae, they are also a kind of bacteria. The blue-green algae is also called as cyanobacteria, students. Very important point. Blue-green algae is also called as cyanobacteria. They are also one kind of bacteria which are having photosynthetic ability, that is they can produce their own food. So bacteria and blue-green algae, they were moved into a new kingdom called as Monera in Whittakers classification. Next, what about fungi? Fungi was also relocated into new kingdom called as kingdom fungi. All the fungi, which included mushrooms, yeast, all the fungi were kept under kingdom fungi. Okay, next, another kingdom Protista has been created. The unicellular organisms, that is amoeba, paramecium, previously which were in kingdom Animalia, they were relocated into new kingdom called as Protista. Now amoeba and paramecium, they are the members of which kingdom, students? They are members of kingdom Protista. This is kingdom Monera, kingdom fungi. Okay, next here, look here. Previously, unicellular algae, Chlorella, Chlamydomonas, these two organisms were included under algae of plant kingdom. Now they have to be changed into new kingdom. So now Chlamydomonas previously, which was in plant kingdom, it has been placed under kingdom Protista because it is a unicellular eukaryote. Next, Chlorella, which was previously in algae of plant kingdom, was kept under kingdom Protista. So these were the changes which happened in Whittakers classification. So is this permanent? No. In future, when the understanding about the living organisms, their evolutionary background occurs, then further there will be some changes in the classification system. Understood everyone? Copy down the students. Students, now let us start the first kingdom under Whittakers classification, that is kingdom Monera. So it is the first kingdom which includes unicellular prokaryotes. This kingdom includes unicellular prokaryotes. All the organisms in this kingdom are unicellular prokaryotes. Okay. And all the organisms are bacteria. It includes all the bacteria. So remember, students, in the last class we have discussed about the simple type of living organisms, the smallest living organisms which is having size of 3 to 5 micrometers. Do you remember? So the same organisms, they are called the bacteria. Okay. So these are the most microscopic or very small living organisms which you can see in the living world. So we have to use microscope in order to see these microorganisms. Then where do they live? So these bacteria are found all around us. Okay, they are found on our skin, they are found in our mouth, they are also present in the air which we breathe, they are also present in the food which we eat. If you take a handful of soil, in that soil we can see some millions of bacteria. Okay, so that means they are present everywhere, almost everywhere they are present. Okay, there is one term called as omnipresent. Bacteria are omnipresent. Omnipresent means they are present everywhere. They are present everywhere. So they are present in soil. They are even present in water. They are even present in the air which we breathe. They are also present inside or outside living organisms, other living organisms. So they are present almost everywhere. You cannot say in this particular place microorganisms, that is bacteria, are not available. They are present almost everywhere. We cannot avoid this bacteria. Okay, so what is the evidence that this bacteria are present everywhere? Suppose if you leave some food material outside, okay, if you don't keep food inside the refrigerator, what will happen? The next day it will start producing some foul smell. Okay, so why that bad smell is coming from that food? Because of the decomposing activity of bacteria. Okay, so this bacteria solely is responsible for decomposition of different food items in our houses. From this we can say that bacteria are present everywhere. We cannot simply avoid them. Okay, next, bacteria are also present in the extreme environmental conditions also. They can live in extreme environmental conditions where human beings or any other animals cannot live. Okay, so what are those extreme conditions? They can also live in extreme environmental conditions. What type of extreme environmental conditions? Like hot springs. So I told you in the very first class in living world, okay, about hot springs. So in hot springs there will be boiling water which is about 100 degrees centigrade. In hot springs there will be water at 100 degrees centigrade. At that boiling water temperature, no other organism can survive, but except some rare kind of bacteria only can survive. So next, they can also be found in polar ice caps, where the temperature will be around minus 20 degrees centigrade. It is less than the freezing temperature. So even that lowest temperatures also some rare bacteria can survive. Okay, and also they are found in salty water. They are also present in extreme salty water. Okay, so in that extreme salt water, no other organism can survive, only some special bacteria can survive. So these are some extreme conditions where some bacteria are still able to survive. Okay, now coming to the shapes of bacteria, different shapes of bacteria. Bacteria come in different shapes. Let us see the 4 different shapes, common type of shapes which we can see in the bacteria. The first shape is spherical shape. The bacteria which are in the shape of spheres are called spherical shaped bacteria. They are called by a term called as cocci. Cocci are spherical shaped bacteria which look like spheres, small spheres under the microscope. Okay, coccus is the singular, cocci is the plural form of this bacteria. Okay, next shape is rod shaped bacteria. The bacteria which appear like rods, you can see here. These are rod shaped bacteria. They are called as bacilli. Bacilli is plural, bacillus is singular. Okay, so inside this rod shaped bacteria you can also find a structure called as spore. some of the bacilli bacteria will produce a structure called spore, which helps in reproduction. It is a kind of asexual reproduction in bacteria. Next, the third shape of bacteria is comma shape. What is comma? It is a punctuation mark, just like a question mark, full stop. There is one punctuation mark called as comma. Okay, comma shape, you can see here. So how we will make comma while writing? We'll do like this, right? This is comma. So these bacteria which are in comma shape, they are called as vibrio. Okay, vibrio. Vibrium is singular, vibrio is a plural. Next, fourth shape, spiral shape. The bacteria which are in the form of a spiral, which are in the shape of a spiral. So how will be a spiral? It will be like this. Okay, this is the spiral shape. The spiral shaped bacteria are called as spirilla. Spirilla represents the plural and singular form is spirillum. What is the singular? Spirillum. So you can ask, what are these structures? These structures are the locomotory structures which help the bacteria to move around. Okay, it is called flagellum. Flagellum. Each single hair-like structure which you are seeing here, it is called as flagellum. It helps in locomotion of bacteria. Some bacteria will have this flagellum and some don't have. Okay, so these are the four shapes of bacteria. I repeat, the spherical shaped bacteria are called as cocci. The rod shaped bacteria are called bacilli. The comma shaped bacteria are called vibrio. The spiral shaped bacteria are called as spirilla. Understood everyone? Come on students, copy down this. Okay students, now look here, I am drawing an example of bacillus bacteria. This is a bacillus bacteria. It is having a cell wall, outer protective covering. This is called a cell wall. So what is the function of this cell wall? The cell wall gives protection to the bacterial cell from the external environmental conditions. So this is the cell wall. This is cell wall. Okay, it gives protection to the bacterial cell. Next, there is a membrane inside the bacterial cell that is called plasma membrane. This is the plasma membrane. This is a thin membrane which is present just below the cell wall. It is called as plasma membrane. Plasma membrane. Okay, so what is the function of this plasma membrane? The plasma membrane, it acts as the outer boundary of the prokaryotic cell and it allows certain useful materials to enter into the cell, such as the food materials. Okay, so that is about plasma membrane. Whereas cell wall, it gives protection to the bacterial cell from the external environmental conditions. Now this bacterial cell, it needs to move. So for its movement and locomotion, there are some whip-like structures. These whip-like structures, they are called as flagella. A single structure is called as flagellum. A single locomotory structure is called as flagellum. Flagellum is singular and plural is flagella. Okay, so this is about bacterial cell. Inside the bacterial cell, there will be a liquid called as cytoplasm. Cytoplasm. Okay, in the cytoplasm there will be DNA. The DNA will be present in the cytoplasm and it will be circular. There will be no open ends. The both ends of DNA will be attached to each other, so it will be circular and it will be folded. Okay, there will be circular DNA inside the prokaryotic cell, and the region where this DNA is present inside the prokaryotic cell, this region we call it as nucleoid. Nucleoid. Okay, understood students? So this is the general structure of a prokaryotic cell. All the bacteria will have more or less similar type of structure. Now you can see the video of bacteria here. You can see here how the bacteria looks like. So when you look under the microscope, the bacteria will look like this. Okay, it will have the prokaryotic cell body and it will have the locomotor structures called as flagella. Okay. Now we will see how this bacteria will obtain its nutrients, its food material. Okay, nutrition. Next subheading, copy down. Nutrition. Nutrition in bacteria. We already studied in Whittakers classification that bacteria will show two types of nutritions. First one is autotrophic nutrition. Autotrophic nutrition. Okay. And under autotrophic nutrition, some of the bacteria show photoautotrophic nutrition. Photoautotrophic nutrition. What is photoautotrophic nutrition? What is photo here? Photo means light. That means they use the sunlight as energy source to prepare their own food material. Okay. So example for this type of photoautotrophic bacteria are blue-green algae. Blue-green algae, come on. What is another name of blue-green algae? Cyanobacteria. Cyanobacteria is the another name of blue-green algae. Okay. So next, what is the second type of autotrophic nutrition? Chemoautotrophic nutrition. Some of the bacteria show chemoautotrophic nutrition. That means this group of bacteria, they obtain their energy from the inorganic chemical compounds which are present in their surroundings. Okay, they derive the energy from which chemical compounds? Inorganic chemicals. Inorganic chemicals such as NO2, NO3, NH3. Okay, this type of chemical compounds will be used by this bacteria for obtaining their energy. NO2 means nitrite, nitrate, ammonia. This type of molecules will be used by chemoautotrophic bacteria, not sunlight. Okay, photo means not essentially sunlight, any kind of light. They use light as a source of their energy to produce their energy. Okay, example for chemoautotrophic bacteria is Nitrobacter. Nitrobacter, example of chemoautotrophic bacteria. Understood? Next, coming to the second group of bacteria which is showing heterotrophic nutrition. Heterotrophic nutrition. The second type of nutrition is heterotrophic nutrition. Heterotrophic nutrition, here the bacteria will obtain their nutrients from external source, from other organisms, from the external source. They cannot prepare their own food. Okay, so this heterotrophic bacteria are again two types. Some are parasitic. That means the bacteria will grow inside the body of other organisms. They live inside the body of other organisms and obtain the food from the other organisms. They are called parasitic. The parasitic bacteria are pathogens. They mostly cause diseases in living organisms. The second type of heterotrophic nutrition, under heterotrophic nutrition, is saprophytic. Saprophytic nutrition. Here the bacteria, they depend upon the decomposing waste material. They does not depend on other organisms for food, but they depend upon the decomposing waste. Okay, saprophytic bacteria, they depend on decomposing waste. Parasitic bacteria, they live in other organisms to obtain their food. So this is about the two different types of nutritions in bacteria. So in the living world, a huge diversity, metabolic diversity is seen in bacteria because of this reason. Because some of the bacteria are behaving as photoautotrophs, some are behaving as chemoautotrophs, some are behaving as parasites, some are behaving as saprophytic. So which group of bacteria will cause diseases? Come on students, tell me. Which group of bacteria will cause diseases? Exactly. That is, parasitic bacteria will cause diseases. Okay, so we'll get different types of bacterial infections, human beings, such as tuberculosis, leprosy, typhoid. So what are all these diseases? Plague. These are all bacterial diseases. They are caused by this parasitic bacteria. They are heterotrophic bacteria. Understood? Copy down the students. Students, now we will see the classification of kingdom Monera by using a flow chart. Kingdom Monera, which includes unicellular prokaryotes, is divided into two groups based upon their ability to survive in extreme environmental conditions and type of the cell wall material which is present in their prokaryotic cell. Archaebacteria. Archaebacteria. These are a kind of ancient bacteria. Eubacteria. Eubacteria. These are true bacteria. Eu means EU means good. Okay, otherwise called as true bacteria. Archaebacteria, they are also called as extremophiles. Come on, tell me why they are called so extremophiles. Since these bacteria, they are able to survive in extreme harsh environmental conditions. That's why they are also called as extremophiles. This is a very important point. Okay, eubacteria, since they have a true cell wall, that's why they are called as eubacteria. These people, they cannot survive in extreme environmental conditions. So these are called true bacteria. True bacteria. Okay, now let us see what are the basic differences in the structure of the cell. Archaebacteria, if you observe the cell wall of this bacteria, the cell wall is made up of some special kind of material which is called as pseudomurine. Pseudomurine. Because of this special cell wall material, this bacteria has capability to survive in extreme harsh conditions. Okay, whereas eubacteria, their cell wall is composed of murine. It is composed of murine. Murine can be also called as peptidoglycan. Peptidoglycan. So peptidoglycan is another name of murine cell wall material. Okay, so this is the difference between the archaebacteria and eubacteria with respect to their cell wall materials. Come on, students, what character makes the archaebacteria to survive in extreme conditions? That is special kind of cell wall. Who is that? Pseudomurine. This is another important point for your NEET exam. Note down this. Okay. Archaebacteria can be classified into three groups based upon the habitats in which they survive. The first group of archaebacteria are thermacidophiles. Thermacidophiles. These bacteria, they survive in extreme high temperatures such as hot springs. Okay. The second group of bacteria, methanogens. Methanogens. These bacteria are producers of methane gas and they survive in absence of oxygen. Okay. Next, the third group of archaebacteria are halophiles. Halophiles. These bacteria survive in extreme salty conditions. Okay. So now let us discuss few points about each of these extremophiles. First, thermacidophiles. Therm means what? Temperature. Acido means acidic conditions. Okay. These bacteria can survive in places where there is extreme high temperatures and also acidic conditions. Okay. What type of habitats they are found? They are found living in, they live in hot springs. Hot springs. So what are hot springs? Hot springs are areas where there is boiling water. The water gets naturally boiled because of the volcanic activity. You can see in the video here. This is the hot spring. See how water is boiling and water vapor is coming out. So this is because of the volcanic activity. Hot springs are found in the areas where there is volcanic activity. In such places you can find this bacteria called thermacidophiles. Okay. So next, methanogens. Methanogens, in the name itself it is there. The chemical methane. These bacteria, they produce methane gas. They produce which gas? They produce methane gas. They produce methane gas. And where these bacteria are found? These bacteria are found in, they are found in lake sediments. They are found in lake sediments. So in the bottom of lake, below the layers of soil, you can find this bacteria. They live in complete absence of oxygen and they release methane gas. And where else they are found? They are also found in marshy areas. Marshy areas. What are marshy areas? These are areas where you can see stagnant water. Stagnant water is present in such places also. You can see, if you have observed any place where there is a stagnant water, some bubbles are coming out in the water. Okay, that is because of these methanogens. Because of their activity, methane gas is rising up in the form of bubbles. Okay, that is because of methanogens. And also methanogens are present in gut of ruminants. They are also found in the gut of ruminants. What are ruminants? Ruminants are nothing but the herbivorous animals like cows and buffaloes. Gut means the intestine. They are present in the intestine of ruminants. Okay. So these are the places where you can find the methanogens. Right. So in the cow dung also we can find these methanogens. And because of these methanogens in the cow dung, methane gas can be produced in biogas plant. Okay. Next, coming to halophiles. Halo means what? Salt. Halophiles are archaebacteria which are found in extreme salty areas. Extreme salty areas. Okay. There will be too much of salt in the water. In such places you can find these halophiles. Example, what is that area? Salt lake. Salt lake. Extreme salty conditions will be present in salt lakes. In salt lakes, because of continuous evaporation of water, all the salt remains in the water itself. Okay. In such extreme salty conditions, these halophile bacteria are able to survive. You can see the video of salt lake here. You can see how all that salt has got deposited at the bottom of the ocean, bottom of this pond. So this is about halophiles. Okay. Now let us move on to eubacteria. Eubacteria are also called as true bacteria and they have cell wall material called as murin, and otherwise it is called peptidoglycan. Eubacteria can be classified into following groups. Blue-green algae. Blue-green algae. These are photosynthetic autotrophic bacteria. They have chlorophyll pigment. By using chlorophyll pigment and sunlight, they can prepare their own food. Okay. These people can be also called as cyanobacteria. Cyanobacteria. Okay. Next, the second group of eubacteria are mycoplasma. Mycoplasma. Mycoplasma are the smallest bacteria. They are the smallest bacteria which does not have cell wall. So in this bacteria, cell wall is totally absent. Cell wall is totally absent. This is one of the important character of mycoplasma, and this bacteria, they cause diseases in plants and animals. Okay. Next, the third group of eubacteria, actinomycetes. Actinomycetes. Actinomycetes bacteria, they are mainly decomposers. They are found living in the forest floor where there is decomposing plant waste. So where they are found living? They are found living in forest floor and they survive by decomposing the plant waste. Okay. So these are the Three types of eubacteria. And the actinomycetes bacteria has cell wall material, a different type of cell wall material which is called mycolic acid. Mycolic acid cell wall material is present in this particular group of bacteria. Come on students, copy down this.